All right, everyone. It is 3 o'clock. We have participants still trickling in, so I'll talk slowly at first and give more folks a chance to join us here in the room. But for those of you that are here, welcome to the IMSMP and Tisch MS Research Center of New York's Quarterly Connect webinar series. I'm so glad that you've joined us for the opportunity to hear directly from one of the most prominent leaders in multiple sclerosis and research and clinical practice, Dr. Saud Sadiq. For those who may not know, Tisch MSRCNY is an independent nonprofit research center dedicated to finding the cause of and cure for multiple sclerosis. We are located in the heart of New York City, and we conduct industry-leading medical research that translates rapidly from the lab bench to the bedside. That translation is facilitated because of our collaboration with the International Multiple Sclerosis Management Practice, a world-renowned clinic offering MS patients access to the best available treatments and integrated support services. The center was just named yesterday to Fortune's list of best workplaces in healthcare and is the only doctor's office in New York of its size to make the list. Together, Tisch MSRCNY and IMSMP make up the world's largest independent MS research center. We're committed to expanding the scientific understanding of MS while redefining patient education and involvement in research, which is why we offer webinars like these. We want you to know what's happening behind the scenes. And there's no one better to answer your questions about MS than the director of the Tisch MS Research Center and founder of IMSMP, Dr. Sadiq. Dr. Sadiq is a board-certified neurologist who completed medical school at the University of Nairobi and residency training in internal medicine in both Kenya and England. He completed a residency in neurology at the University of Texas and a fellowship at Columbia University right here in New York. He has served as both director and chairman of the Department of Neurology at St. Luke's Roosevelt and held faculty appointments at Columbia University and Albert Einstein College of Medicine. He has over 100 publications, has supported the early careers of 140 medical students through the Tisch MS Research Assistantship Program, and is widely acknowledged for his pioneering work in developing intrathecal therapy for patients with MS. Dr. Sadiq is also one of the few MS specialists who has a full-time clinical practice in addition to a lab-based research program. He's been named a Castle Connolly Top Doctor every year since 1996, and he mentors emerging neurologists and neuroimmunologists that are interested in multiple sclerosis through a fellowship program here at the center. My name is Sarah Bond, and I lead communications at the center, and want to kick us off with a bit of housekeeping. Today's webinar is being recorded and will be sent to you on Monday. So if you'd like to watch it again or share it with friends or colleagues, feel free to do so. We are prioritizing questions today that were pre-submitted via the Q&A form that was listed on your registration page. And I can already tell you that due to the enthusiasm of your contributions, we won't be able to answer all of the questions today, but they will help inform future content. So without further ado, since we have so much to get through, let's dive right into these questions that were submitted by you. Dr. Sadiq, I'd like to start us off with a few research questions. The first one being, what do you feel is the most promising future therapy for primary progressive MS? And how far away are we from remyelination?
All right, so that's a difficult question because it kind of goes into two areas that are not related. I'll take it as what the question means is what are current immune-based treatments for primary progressive MS like ocrelizumab, which is approved for primary progressive MS. Are there other agents like that? And then the remyelination is a separate thing, which is regenerative medicine. That may not just be for primary progressive, it may be for any form of MS in which patients have disability. So there are no, as far as I'm aware, and I could be just ignorant, there is no real promising treatment being developed for primary progressive MS, in my opinion. Now, I'm a little bit outside the pharmaceutical industry, and so they have a lot of promising agents that they say are promising, but I don't necessarily share their views. And the reason is that we have to first work out what are the mechanisms of primary progressive MS. And then once we know exactly how that happens, then we can really develop promising treatments. Now, just as a way of comparison, natalizumab or Tysabri is a promising treatment for relapsing remitting MS. So when we did the initial trials that I was not involved in, except at the later stage, the results were just immediately evident, and you didn't have to do very fancy statistical work to know that it was effective. So in that way, there's no really highly promising drug for primary progressive MS, but we are doing research in it in the field. Jamie Wong in my laboratory, in our laboratory, I should say, has done some very innovative work and is a new model for primary progressive MS. And we are in collaboration with a place with a research center in Holland and also one in California, that they're looking at molecules that may develop into promising treatments, but it would be premature to say at this point. And just finally, before I leave the subject, we do know that antibodies in the spinal fluid are responsible for some of the pathology that we see with primary progressive MS. So as a research program, once we know what the antibodies are doing, we will be able to, and how they are causing the problem, we may be able to develop effective treatments. The remyelination is a separate question. I don't know if I should answer it. I'm running out of time. But that's our phase 2 trial that we did, and I'll talk about that a little bit further along, I think.
Yeah, okay. So just looking at the first part of that question, you're saying that from a pharmaceutical perspective, there are no future pharmaceutical therapeutics that you're really excited about.
Well,
But the most
I don't want to be
but that the most
pessimistic. There are a lot of
effective
agents
route.
being developed. I just don't know how good they'll be.
Gotcha. Yeah. So that's why you're saying the more effective route is to figure out the true mechanism of PPMS. Because if drugs are being developed without the true mechanism being known, how effective can they be really? So in our lab, you're focused on isolating the mechanism of PPMS. And I know that we're able to do that because we have a novel model of PPMS that's been developed by Dr. Wong. Is that a fair summary of what you've said?
Yes.
Okay, and let's go ahead and address that remyelination question now. Folks are wondering, is remyelination possible, and what's the time horizon like for that?
All right. So remyelination is not the only cure for whatever. You need nerve and myelin repair, and there is scars formation in the brain and spinal cord when you get damaged. So when you plan for a repair strategy, it's not just remyelinating. And the people who are taking, all sorts of medications that show remyelination in platforms, people have been taking drugs that are for diabetes and so on, like metformin, and think that they will remyelinate. That's not really going to be effective. So we've been concentrating on stem cells that Dr. Harris has worked in my laboratory, in our laboratory rather, and we did a phase 1 trial, which was widely publicized, and a phase 2 trial that was basically using stem cells that were derived from the bone marrow. And the problem with that is obviously the bone marrow wasn't made to make stem cells for MS. They were made for blood stem cells. So we took a very small proportion, a very small part of those cells that are called mesenchymal stem cells, expanded them, and then made them into brain-like progenitor or pre-brain cells, and then injected them into the spinal fluid. And all of you are aware, we published those two papers that Harris was first author on 2018 and 2024, phase 1 and phase 2. And then we decided at the end of that, that although there was some benefit, it wasn't what we had hoped for, at least what I had hoped for. But we are now developing some new strategies based on technology that has gone along, which may involve us to reprogram genetic material found in blood of everybody, and basically reprogram some of the material so that we can use embryonic-like stem cells. They're called induced pluripotent stem cells, but iPSCs. But so these have a lot more potential because they are not so age dependent, and we are hoping that we can do all the necessary laboratory work that we need to assure their quality and other genetic markers to see if they are appropriate and if they have the potential of developing into cells that can remyelinate. And I think once we start that trial, hopefully in a year, that will be very promising. The one positive thing about that is we have used similar technology to make mini brains and mini spinal cords from this kind of cells. So I'm hopeful. We could never do that with bone marrow cells. So I think based on just on that, and we have published on these organoid work that was done by Nicolas Daviud in our lab from France, that I'm optimistic about, accomplishing a real repair strategy that would be effective in the next few years, in the next 2 to 3 years.
Okay, so let me try to condense this. We hope to launch a new study in about a year that's focusing on this new type of stem cell that we think will make remyelination more possible. That's absolutely the goal. That's what we're all working toward. And this new type of stem cell that we're working with, these iPSCs, these embryonic-like stem cells, are really just a technological revolution. Dr. Sadiq and I were speaking to some folks earlier this week, and we compared working with bone marrow stem cells in this context to these new iPSCs, like the difference between a monk copying a manuscript by hand and the advent of the printing press. The technological jump and scientific capability jump is huge. So now we're at the point where our scientific capabilities have caught up with Dr. Sadiq and Dr. Harris and Dr. Wong's research ideas. And now we're able to execute on that research more effectively because we have more powerful raw material. And Dr. Sadiq, let's just differentiate one more time the difference between a bone marrow stem cell and an iPSC. If I'm understanding it correctly, a bone marrow stem cell is already specialized, and an iPSC is something that we create in the lab. We take out patient blood, we perform some processes on it to turn it into something that acts like an embryonic stem cell. Of course, true embryonic stem cells are harvested from embryos, but these don't involve actual embryos at all. They're able to be created from adult blood, so they're unique to the patient. And then once we have them reprogrammed, they're sort of like this blank slate. They're a master key that can turn into any type of cell in the body: nerve, brain, muscle, blood, whereas bone marrow stem cells are only adult stem cells that can be blood stem cells. Is that correct?
Just the last part. Well, the bone marrow cells are slow. They grow. Bone marrow cells used to die after 4 or 5 periods of growth. And Dr. Harris was showing me some graphs the other day, and these cells just keep multiplying and producing, and you can use many cycles of replication, and they're still good. And so, yes, there's a tremendous potential with these cells to do what we want them to do. It's much better, much easier. At least in the laboratory so far, I've been very excited. We'll have to see what happens in live, once we start the trials.
Excellent. Well, I know we're all looking forward to that trial beginning in about a year. And then, as you said, science takes time, but perhaps 2 or 3 years after that, we'll have some results and exciting findings to publish. Moving on to one more research question before we switch gears a little bit. How are participants selected for clinical trials, both here at the Tisch MS Center, but then just more broadly? Walk us through what recruiting participants looks like for clinical trials.
So when we are ready to start a trial, we also meet with the FDA, and usually I don't know if the FDA has any employees left, but if we have an FDA meeting, Dr. Harris and myself will usually meet with some regulatory officials, and the clinical trial design is then decided that we are going to target which type of population of patients, what's usually between 18 and 65 or even 75 or whatever. And then we can decide whether we want to concentrate on people with a certain amount of disability or active disease or not. So there are the clinical design is decided upon, and then we have to communicate with the statistician to make sure that we have enough numbers so that if we get results, we can interpret them and draw scientific-based conclusions from that data. However, any patient with some level of disability, I think, would be a suitable candidate. We'll have to decide. I usually try, in the last trial, we had secondary progressive and primary progressive patients, but all of them had it to be ambulatory because we were looking at walking speed and so on. So we probably will have a similar kind of design, but and it also is really dependent on funding and how much we have availability, because each patient there's a significant cost attached to a large study. Normally, we don't have to even recruit anybody from outside the center because we have just such a high demand among our patients. So I don't actually recruit anybody from outside centers unless they're already my patients. And if it works, of course, it will be made available to every center in the United States if they are willing to do it. If it works, that'll be a good problem to have.
Great. So you speak with the FDA, you speak with a statistician, and then there's some diagnostic criteria that's used as a filter based on the study design that's been decided with those two parties. Does that make sense? Moving on to some practice-related questions. We had one that was submitted. How do you decide the course of actions for RRMS patients with DMTs? And do you think that infusions last longer than oral DMTs?
So the decision for treatment is based on the patient's individual. There's a lot of individual decisions you have to make as the patient's age, what are their priorities, how much disability do they have to begin with, and where they are. The oral drugs generally for relapsing remitting MS are not as effective as the intravenous-based drugs in efficacy, and also some of them are more problematic if there are female patients who are of the childbearing age. So for most RRMS patients who are between 20 and 40, and the typical patient is a female or then you would probably take into account that she may want to have a pregnancy and so on. So I usually, if they can, would go for an intravenous drug, a medication, and I try not to disrupt the immune system if I don't have to. And so usually if the antibodies to JC virus that's associated with PML in patients who take natalizumab or Tysabri, if they are negative, I would usually first start off with natalizumab which I still think is the most effective treatment for relapsing remitting MS with the least amount of side effects. The oral drugs are mostly more broadly immunosuppressive, and when you stop them, if somebody wants to get pregnant, there's usually a rebound associated with it, which can result in relapses. And so my first choice would probably be the taluzumab, and my second choice would be an anti-B cell therapy like ocrelizumab.
Thank you, appreciate that. Another question we received was, I'd like to know Dr. Sadiq's view on RRMS patients quitting therapy after they turn 62. Similarly, are DMT side effects for patients over 65 more harmful than beneficial? So can you talk a little bit about the aging process in DMTs?
Well, stopping treatment doesn't make sense to me. I think if somebody has a disease that needs treatment, it needs treatment. I don't know what that 62 age came into. Some people wanted to study it. I know some of my colleagues have said there is no need to treat them, but I've had patients with relapses well into their 60s. I've had patients in over 70 have relapses, and previously were walking and then stopped walking after that. So I think it's a little foolish to make generalizations like that. In my opinion, I think you individually treat a patient. Nothing happens when you're 65. I mean, not nowadays. I mean, now I have to, for full disclosure, I mean, I'm over 65 and I wouldn't stop taking a blood pressure pill if I was taking one just because I'm over 65. No, you treat the person individually, and if their disease burden was high and somebody was progressing, I mean, and they've stopped progressing on treatment, I would leave them alone and just treat them. One concern would be side effects at over 65 of suppressing their immune system, but you can you can manage a patient. It depends whether they're smokers or not, if they have kidney function problems or liver, heart problems. I mean, there's nothing mysterious about MS. You treat them like every patient is an individual, and you make these individual decisions. People look for herd statistics, and they want automatic, studies to be done, and that we can say, okay, after 62, I don't have to treat the patient. And that's just physicians trying to be lazy, I think. They don't want to spend time to look at an individual. These are individual decisions.
Mm-hmm. And regardless of a patient's age, if they're experiencing side effects that are emergent and difficult, that's something that you treat right away, whether they're over 65 or under 25. So the idea of side effects being more harmful than the drugs are being beneficial, that's something that I know here at the practice you would address immediately, regardless of age. Can you let us know the age of your oldest patient, Dr. Sadiq?
I think someone, oh yes, he's about 97 now.
97.
Yes.
Yeah.
I don't
So,
he's the
so
oldest.
yeah, we had no
And
worries there.
he's still ambulatory. He uses a walker, but he's still good.
That's amazing. Another question based on clinical practice. Do you think BTK inhibitors will be safe and effective? Why
I
don't we
don't
start with explaining what a BTK inhibitor is?
know. Anyway, BTK inhibitors, there's been a lot of trials. There is this whole, the pharmaceutical industry has brought up this concept of smoldering MS that, so they can make a new class of drugs that will help them. And they were supposed to interfere with B cell function because it inhibits an enzyme, glutathione kinase, that's why it's called BTK. And then they were found not to be as effective as B cell therapies. So then they had liver toxicity, and the effects are all over the place. And I was talking to Dr. Stark earlier, and he said the United Arab Emirates, which is not usually considered like this, well, whatever, They just approved a BTK inhibitor for MS, for non-progressive MS. So I think I don't know if they'll be safe. They have problems with the liver. There's been some other toxicity. The therapeutic effect has been unimpressive to me, at least. I've just looked at what the pharmaceutical industry puts out. I was not involved with the clinical trials with BTK inhibitors, so I think the jury is still out on it. We'll wait and see. They're not going to be a huge game changer. I think there'll be new drugs, so people like to try new things, but um, certainly I don't think they're as effective as Tesabri or B-cell therapies. Now, my colleagues who are all over the place and usually funded by the pharmaceutical companies will say, no, 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 this addresses the smoldering MS, and but there's no proof of that, you know. So it's just what they want to talk about it so they can say that it's effective. What inhibits glial cells in vivo in a live person, we haven't really shown. At our center, we've used intrathecal methotrexate, which nobody would use because it only costs $5. And nobody makes money, and it's no patent. So, we'll wait. I'll say I think the first drug's going to be approved, tolebrutinib, in about a month, and then we'll see. But there is, I think, significant toxicity with some of these drugs, so we'll have to wait and see. And the efficacy or the effectiveness hasn't been like, wow, you know, we have to use these. I wouldn't be. I'm not waiting with bated breath to use them.
Yep, makes sense. Speaking of pharmaceutical funding, let's go back to the research side of things, because we had a somewhat related question. A few folks were curious about what effect the recent domestic policy changes, like what effect they will have on research, the big beautiful bill, if you will. What effect will that have on research, both here at our center and elsewhere writ large?
Well, luckily for many years ago, I decided to leave the traditional academic route, which is Columbia University, and then St. Luke's Roosevelt, which is now Mount Sinai, mainly because they're very bureaucratic. There's a lot of wastage. A lot of money goes for indirect costs, and the priorities of the institution change, and you tend to publish work that's safe and publishable to get promoted, and there's a whole culture where you waste a lot of money, and so we've become independent of that. We opened the center to remove all these barriers to rapid research development. So luckily, the beautiful or ugly bill or whatever you call it doesn't affect me or at our center. I think at certain academic centers, there has been funding changes, but I think good work is always possible. There is enough innovative work and innovative resources, and a lot of the really good work is being done by pharma. You know, they do basic research like at Genentech. The real scientists are totally not connected with drug development. They do a lot of groundbreaking research work, and there are many institutions that are not dependent on this type of funding. So I don't think it'll, in the long run, have a big effect, although there are a lot less research positions. So eventually, maybe there'll be less scientists, but maybe it will just allow the really good scientists to survive. Difficulties like this are always good. You know, it's survival of the fittest. So I don't ever worry about this background noise.
And that is a huge advantage to the way we fund our work, is that we're insulated from policy changes. And we are able to maintain a very laser focused and tight research goal Whereas when you're with a large university or have the government funding, the goals get diffused a bit more and they can splinter off into various pathways, whereas we are solely focused on finding the causal mechanisms that you mentioned earlier and an eventual cure for MS, which I think is pretty interesting. Another question we received on research: what other global research specific to repair and improving disability, other than the research being performed here at Tisch, has sparked your interest? And
for repair, I think there are a few laboratories that do good work in Cambridge in England, and some scientists in Israel and also in Italy. I don't know if they're ready to do clinical trials in MS. There's a whole platform of medications that are being used, especially in California, that are really being pushed, and they're antifungal agents and anti-diabetic agents and so on. And they have used this platform where they look at cells that make myelin in a clinical set, purely in the laboratory, and develop those agents. That I'm not very, I'm not convinced that they will ever work, but I think these other places where some stem cell work is being considered, the ones I mentioned, may be of interest, but nothing has really come in the literature recently that is really getting me excited. I just reviewed an awful paper that I rejected for stem cells done on some journal, but so I'm not trying to be pessimistic, but I'm just telling you, I'm just waiting for other people. They haven't published what at least maybe they're just developing things.
I mean, that makes sense. If it interested you, you'd be doing it. I think it might be helpful for folks listening to understand what would make you reject a paper. So, peer reviewed, all of these great journal articles are peer reviewed, meaning folks like Dr. Sadiq and Dr. Harris read articles and provide feedback on them. So they're not just published willy-nilly in a scientific journal. What are some things about this paper, specifically, Dr. Sadiq, that put up the red flag for you, that made you think, this isn't quite up to snuff?
I probably should have brought it up, but anyway, this was a paper done by Europeans, a European center, and there were several problems with it. It was a small study, it was poorly designed. The conclusions were not valid based on the data, and I think their cell manufacturing technique was probably at fault because they had some adverse effects that were associated with spinal fluid injections of stem cells. And I've done a lot of spinal injections, both with spasticity drugs, pain drugs. I've been doing intrathecal therapy since 1991, and stem cells with Dr. Harris, and we've never had those type of side effects. So my criticism was based on that. There was also a very small study with a lot of larger oversized conclusions. So I just suggested that perhaps it should be revised, or certain things should be rewritten. But that's common. I mean, a lot of my papers get rejected as well in first time you submit them, or at least be asked to get revised.
Yeah, I think that it's helpful for everyone to remember that that is part of the publishing process. We design these studies as meticulously as we can. We collect data, we spend time in analysis, and then the manuscript is written and submitted. But then there's always some amount of revision that happens. That can be one round, multiple rounds, or in some cases, the paper isn't able to move forward because the design may have some inescapable holes in it. So thanks for highlighting that process for us a bit. Another question we received was, can you explain the research on stress and multiple sclerosis?
So stress is not one thing, you know, stress is very, you know, you can't really do appropriate research into things like this, diet, stress. There's a lot of things that look good to do, but they're so varied and wide, you know, people have such differences. What one person can find stressful, the other person cannot. So I try to look at the whole way you live. Are you in a toxic environment? Is it your spouse, your work, whatever? All of those things need to be for healthy, for the best life you need to have absence of stress. And that's a goal, I think. You get better outcomes, better treatment, and so on. And so if you can eliminate stress, whichever way, and I don't mean you eliminate your spouse if they're causing stress, you can, divorce, or you can work on a marriage counselor, although that doesn't really ever, I don't know if it, well, I probably work on a counselor or whatever, but you can get therapy, you can do yoga, you can pray, you can do all these things, Pilates, walk in Central Park or walk in some non-toxic place, environmentally non-toxic. You can go and see the Northern Star or lights or whatever moves you to feel calm, and you can drink green tea or blue tea or whatever it is. But basically, stress reduction is good, and you can find a happy place. That's good. And if you do what you want, so I have no stress in my life. I don't worry about anything. You do your best, you succeed, you thank God, you go to sleep, you feel good about life. Everybody you work with, you should encourage them to do better. If they criticize you, you should take it as constructive criticism. Try to be better. You try to do best for your patients. And if things are well, things move along. And if you're stressed, then something is not you're not doing right. I always look at myself. If I'm feeling stressed, instead of blaming somebody else, look at yourself and try to improve yourself and try to improve your own environment. And you'll find that basically there's nothing you're controlling. So if you're not controlling anything, why should you stress about it? Just try to do the best you can. And there are very few things in life you can control anyway. And but toxic stress is not a good thing to have if that's the case. And we have therapists and social workers who help patients, and you should seek help if you feel like you're being stressed out.
What changes have you seen in patients that have been able to better manage their stress throughout their time at the practice? What do you notice once people get their stress under control?
So, so stress management really helps with people's just feeling good about themselves. And I think there is a therapeutic milieu. I mean, our center is designed, like a spa, so that if people come in, just feel a sense of well-being. You go for infusion therapy, it's very calm, everybody's on time. Everything that would cause aggravation to a patient, we try to eliminate. Waiting time, how you polite, you're respectful of their time, you address their needs, you never rush them out. They feel like they're the center of your universe when you're with them. Those are all stress reduction aspects. But we have social workers, we have a nutritionist, we have a naturopathic doctor, and also I think for patients, what they should look at is in an ultra-Orthodox Jewish patient, that's a large population of a large percentage of my patients. We always have a different kind of stress management for them, which is mostly religious based, but there are also people who are not of that conviction with God, and so they do, Pilates and all these other things, that yoga and so on, and that those things seem to help people, deal with. These are all designed to be stressful, even just bicycling and um, walking and all these type of exercises and that people indulge in. I have my own little thing, and you can meditate, you can do whatever that helps you reduce your stress. And I think that's helpful. It's part of the treatment of a patient.
And it seems like it improves patient resilience which of course is important when it comes to managing symptoms and staying on top of appointments, all that sort of thing. We had a question about dysautonomia. Is there a connection between dysautonomia and multiple sclerosis?
So dysautonomia, for those who don't know, there is a central nervous system, which is the brain and spinal cord. Then there's the peripheral nervous system, which is the nerves in the arms and legs that make you move your fingers and so on from the spinal cord onwards. And then there is an autonomic nervous system, which is responsible for your heartbeat, your bladder and bowel function. Your GI system is working while you're talking this website. And that's considered autonomic nervous system. And people usually with MS who have spinal cord lesions will have bladder bowel problems or sexual problems and with function. And then there is some other aspects that people will say are autonomic nervous system functions like fatigue and so on. Pure autonomic dysfunction, autonomic nervous system dysfunction actually is a slightly different definition by health. That's your blood pressure control is bad. You have syncopal attacks. When you get up suddenly, your blood pressure doesn't keep up. Your heart rate variability is high, and they can have certain problems with just their control, and could involve temperature control as well, can be all over the place without infections. So pure autonomic dysfunction is very rare in multiple sclerosis, although my colleague, Dr. Williams, seems to see more of it. But I've seen it occasionally, but it's not that common. But dysfunction of autonomic nervous system, bladder, bowel function, sexual function, and so on, that's fairly common, but I classify it slightly differently. So there is a very sort of a connection between autonomic nervous system function and MS, but dysautonomia, the pure term, is less common and is less of a, and it can occur, usually occurs in not MS, but other, just on its own, with these, some other syndromes like Scheidegger syndrome and so on that can cause that, and even multi-system system atrophy and other nervous system diseases. All right.
Thank you. We had someone write in that their son was just diagnosed with MS, and they were diagnosed 19 years ago with relapsing rabbinic MS. If MS is not hereditary, how does this occur? And there were several other questions of that flavor asking about MS within bloodlines. So can you talk about that a little bit?
So MS is not inherited like another, like hemophilia, which is a blood clotting disorder is, or where there are certain genetic diseases that occur. MS is not a genetic disease in that monozygotic twins that share the same genes are not 100% concordant. That means they don't both always get MS, only about 30% chance that they will have one will have MS on one, and they have the same genetics. But there are families in which you get a higher chance of you getting MS. And really what it comes down to is if you have no parent with MS, your incidence of your chance of getting MS is about 1 in 350. Some people may say 1 in 400. And if you have a mother or a father who has MS, then your chance could be 1 in 100, or some people will say 2 in 100. And if you have multiple members, then it's slightly higher. So there is a genetic predisposition, but it's there, and it's just like, there's a higher chance that you will be a terrorist attack in New York rather than if you're living in North Dakota somewhere. But it's just a genetic predisposition, it's not inherited. And yes, I do have parents with kids who have MS, and they're both my patients, but the majority of my patients don't have multiple family members with MS.
Thank you. This next one is a bit of a theoretical question, and you sort of touched on it earlier in our discussion with pharmaceutical companies reclassifying this smoldering MS situation. They asked, can you provide your perspective on the discussion within the MS community around classifications of MS diagnoses, relapsing remitting, PPMS, etc., versus viewing MS as one disease continuum?
So MS is probably not one disease, at least data from, when I started in the field, we didn't have NMO, we didn't have MOG. We used to call it, like, people weren't quite sure what that was. Somebody used to call it DEVIC. And now I think personally, relapsing remitting MS, whatever it is, evolving to secondary progressive MS is the same disease. It's just different stages of the disease. Which is, so relapsing remitting, if it's inappropriately treated or not actively treated, or sometimes it just happens, it can evolve to a secondary progressive form. Primary progressive MS, I think, is a slightly different disease as far as causation is concerned. And certainly our laboratory work, particularly work by Jerry Lin, but more importantly in the experimental model with Jamie Wong, and even with the organoid work that Nicola Daviut started and Priti Kalpana is doing, probably suggests that the way primary progressive and relapsing remitting may be two different diseases, because they tend to have different proteins that are expressed, or that the antibodies in primary progressive, and where primary progressive affects the spinal cord and secondary progressive may affect the brain, or relapsing remitting. So we're still trying to sort it out. And this happens with diseases where the cause is not known. Until you can really identify the cause of a disease, that's when you really know for sure whether it's one disease. But it shouldn't make people nervous or special. I mean, if you just take something very simple like hypertension, which is high blood pressure, you can have high blood pressure for many reasons. And then, so you can treat it similarly, but it's different things in different people. Somebody might have renal kidney disease and then get hypertension. Other people just have a genetic predisposition. Other people have dietary and weight reasons for having high blood pressure. So, and it could be many other causes of it. So I think with MS, there's no, but I think you can't, I'm more of, you have to, when you make treatment decisions, they should be based on an individual and many other individual characteristics that go into play, not just saying, oh, you have relapsing remitting, this is what you should do.
Mm-hmm. Can you say a few words on why it's so difficult to find the cause of MS, why that's been so elusive for researchers over the years?
Well, it's well, first of all, multiple sclerosis only occurs naturally in humans. So once you have a human disease that only occurs in humans, it's hard to study. If you want to study hypertension, you can just, in a mouse, you can just clog up the renal artery and you get hypertension and you can study it. In MS, it's hard. There's no real good animal models. So our goal in our laboratory has always been to try to study a human disease. Now, you don't get brain from patients until, of course, they have died, and then it's different. Even if they donate their brain to research, it's a dead tissue, it's not a living tissue. So there were several problems. Then the other issue is, for many years, we've known that Epstein-Barr virus, for example, plays a role in the cause of MS. However, Epstein-Barr, again, is a virus that affects only humans, doesn't affect mice. So how do you study it in a human being? You can't infect it and then see what happens, because and then there's 90% of humans get EBV or Epstein-Barr virus, but only 1 in 350 get MS. So you figure why does some people get MS with Epstein-Barr, and the vast majority of people don't. So to study that, you need to kind of have a human model, see what the virus does. The virus also becomes latent, sits and goes to sleep. And to study viruses in general, they need to be awake so you can, or be active, and then you can study them. So there are several problems, and I can go on and on and on, because this is all I think about all day. So there are a couple of things that have happened recently that have got me excited. Roberto Alfonso, who's in our laboratory, is a virologist, has been working with Epstein-Barr and trying to figure out, is there a separate cell that it infects that causes those patients to have MS? So if it was a brain cell, because normally Epstein-Barr sits in B cells or immune cells, but if it can get into a central nervous cell, then it would explain why the immune system is directed against the central nervous system and cause demyelination. But then to study it, he couldn't really put the EBV virus in mice neurons or mice nerve cells or rat cells because they don't get EBV naturally. But then this new way of making stem cells that I was talking about that allows us to make mini brains, which are called organoids. So, and then we can now use that to infect Epstein-Barr virus and see what would happen. And that's a difficult challenge because these brain organoids don't have an immune system. And so Dr. Kalpana is trying to figure out how to get an immune system into the organoids. And then we can use EBV and then we can try to eliminate it. So the cause of MS, and that may be just one aspect. Then Jerry Lin has been working on these antibodies that come in spinal fluid that we don't know why and how they arise and what they're directed against. And that's been 20 years of work. So it's right around the corner, but it keeps moving. So it's like a mirage in a desert. I always keep looking for the water, but it's always eluded me and many other people. So one day we'll find it. That's how I think everything works.
Yeah.
We are actually closer to primary progressive MS to finding the cause than with relapsing, but which is something I wouldn't have predicted. But it will happen. It just takes time.
Mm-hmm. So it sounds like the biggest roadblock has been that until recently, there's been no one-to-one models to study these diseases. There's always been something about them that's not quite right and indicative of what the disease looks like in humans. But the technology is catching up, so we're getting there. We have time for about 2 more questions. I'll ask one more about research. Does Tisch MS do work focused on the gut microbiome, given the latest research about gut bacteria differences between MS patients and people without MS? And why or why not?
So we have Dr. Bates, who's our naturopathic doctor. She's always talking about and working with the gut microbiome. The gut microbiome and the paper that was published about the 2 bacteria, people have asked me, what do you want to do? Should we take antibiotics? But this is not the way you think about it, because if the gut microbiome is involved in initiating the disease, once the disease has already started, taking antibiotics won't help. And also the gut microbiome is got so many bacteria. We have more bacteria than human tissue. And so sorting it out is very difficult. I personally haven't been involved with the gut microbiome data. I do think that there is certainly infectious triggers that interact with Epstein-Barr virus. At least that's one of my hypotheses, that allow the immune system to start attacking the central nervous system. But whether that's a bacteria that's in the environment or in the gut microbiome, I don't know. So this answer is not very satisfactory. I'm sure you have realized, who are listening, that I personally don't do gut microbiome research. Although Dr. Bates does work with you on that, and does tests to see what kind of therapy and even sometimes antibiotics that she can use to make you feel better. And then usually these are quality of life changes like fatigue and well-being when people ache and just generally have malaise. Those type of symptoms do improve when you see somebody like who works on the gut microbiome.
Mm-hmm. And I think that goes back to what we were saying earlier, that we have a really focused research goal, and we stick to the things that we see as the highest potential avenues for creating real change. rather than having a broad and shallow research focus. We have a few really narrow, deep, high potential, heavily researched areas that we work in. So that makes sense to me. Our final question for you, Dr. Sadiq. What is one thing that you think MS patients commonly misunderstand about the disease? Whether they're newly diagnosed or 2 decades into their diagnosis, what's something that you think MS patients commonly misunderstand?
Most patients are like students. So if students misunderstand something, it's the teacher's fault. So I find that MS patients don't misunderstand anything. If they misunderstand something, it's the doctor's problem. When you first meet with a patient, you talk to them about MS, and there should be everything that they have that are misconceptions or so on, you try to teach them and you correct them. And if they still have misunderstanding at the end of the interview, then you have failed as a doctor. So there are no misunderstandings. There are only doctors who cause patients to have misunderstandings. You need time to take with your patients. That's what you should do. So it's our fault when they have misunderstandings.
Before you have those conversations with them, what misconceptions do they come to you with?
Oh, I'll be disabled, I will be this. I mean, this is not misunderstandings, that's just fears. They're not misunderstandings. It's the fear of the unknown. So when you make it known, when you know something, you don't fear it. So it's the fear of the unknown, that's it probably. But it's not a misunderstanding. I would classify it as just not knowing. I mean, ignorance leads to fear. Fear leads to, all the bad things that happen, So when, I think, once you can educate patients and patients get educated, then they would not. So misunderstandings are always, I think, a lack of knowledge. So that's probably, if you want to get a patient, if you want to answer the question simply, what is the greatest misunderstanding? It's just a lack of, it's a lack of knowledge about the disease. It's an exciting time to be in MS. There's a lot of research, a lot of work has been done. I've been in neurology for over 50 years, and in ALS, nothing has happened during that time. From Lou Gehrig to now, we're still trying to figure out what to do. And in MS, the whole therapeutic array of drugs has changed. Patients' outlooks are much better, and we will strive forward until we can face a future without MS.
It is a very exciting time to be associated with multiple sclerosis practice and research. That is all of our time today. Thank you so much, Dr. Sadiq, for sharing your knowledge with us. And if you're interested in more webinars like this, our next Quarterly Connect webinar is on November the 14th. That one will focus on navigating the holiday season with MS, and we will have all of our neurologists as well as specialists from across other fields of MS practice here to talk to you about the different facets of navigating the holiday season with MS. If you'd like real-time updates from our research lab, you can follow us on social media at Tisch MS, TISCHMS. And if you'd like real-time updates from the practice, you can follow us at imsmp.nyc. I hope to connect with you virtually. Thank you again all so much for submitting your excellent questions and for taking time out of your day to commit to science and learning more about multiple sclerosis. Dr. Sadiq, thank you so much. Wishing everyone a healthy and happy weekend. Thank you.
Thank you, sir. Thank you, everybody.